Okay, I think we can start. So it's a huge pleasure to introduce Professor Stein Aerts today for the ERGA BioGenome Analysis and Application Seminar. Stein leads the Laboratory of Computational Biology at the University of Leuven and the Flemish Institute of Biotechnology here in Belgium. And he's also directing the development of the new VIB Center of AI. Stein's research is truly at the forefront of deciphering the gene regulatory code and how it drives the dynamic changes in cellular states. And this in a multitude of both tissues and species. So I'm very excited to give him the word and hear about his most recent developments and findings. Thank you, Stein.
Thank you so much, Steven and Christian and everybody for the invitation. I'll share my screen. Does this come through? Yeah. So thanks again to ERGA for the opportunity to give a presentation here. My lab is focused on, like Steven said, a question of can we decipher the genomic regulatory code to understand how the genome encodes the information to yield spatiotemporal expression of all the genes in the genome, like this gene here, alpha-tri, that is expressed specifically in the gut of the fruit fly. And what it means to decipher this code is basically to identify, to find and characterize all the regulatory regions, enhancers, promoters around target genes, to find out what are the binding sites of transcription factors, what are the transcription factors that bind there cooperatively, which ones are activators, which ones are repressors, and how do these different enhancers and promoters integrate to yield a certain mRNA initiation rate at every target gene. So in the future, we can envision to have that information, like a full transcriptome for every cell of an organism. This is spatial transcriptomics data that we recently obtained for the fly for 100 genes. So we are not there yet to obtain the full transcriptome of all the cells, but we can already approximate that with single-cell RNA-seq, as I will show. And ultimately, then if we have this full output of the regulatory genome, then the idea is to reverse engineer this to build gene regulatory networks and to understand the regulatory logic, or reciprocally to train AI models on this genome sequence to predict this as an output. As I said, we can approximate already all the transcriptomes of all the cell types by single-cell RNA-seq, and this we did in the Fly Cell Atlas. This was published in 2022, and with a consortium of about 40 different Drosophila labs, but led by two people, Jasper in my lab and Hongji, who's now at Baylor in the lab of Li-Chun Luo at Stanford. We're leading this analysis, and for that, we obtained single-cell nucleus transcriptomes from the head, from the body, and from 15 dissected tissues, male and female, and after FACS to clean up the debris, performed 10x and SmartSeq2 to yield the Fly Cell Atlas with 580,000 cells. In my lab, we also built, led by Chris, a software called Scope that allows us to analyze these single-cell data remotely with all the different expert labs on the tissues to basically find and annotate all the cell types back. And then using marker genes, the researchers in the different labs could annotate these clusters here using a controlled vocabulary from Flybase. And that led to 252 annotations from all the different experts, from the experts on the fat body, experts on the heart, on the Malpighian tubule, here shown in a zoom in, where most of the clusters of cells are annotated as cell types that were known from decades of developmental biology with marker genes in the fly. And before going further with asking what are the underlying gene regulatory networks, just a shout out to the Biodiversity Cell Atlas. So having this Fly Cell Atlas expertise, it could be applied and will be applied to a large number of species. This is led by Arnau Sebepedros from CRG. The initiation of the BCA was in May last year, with all these people here involved in the coordination. And in the BCA, the idea will be to generate such atlases, as I showed you for the fly cell atlas, for hundreds of species together with their genome. So the Earth BioGenome Project was also involved in this meeting, and I think this could be relevant for you guys at ERGA to also associate with. So going back to the fly cell atlas, if we want to know for each cell type what are the underlying regulatory principles, the first simple thing we could do is to look at expression of transcription factors. So all these, each row here is a TF, and here you have a number of cell types. And even though TFs are usually expressed in multiple cell types, each cell type does have a unique combination of transcription factors with some smaller number of, let's say, master regulators. And by linking them, these names of the TFs to these different cell types, you already have an idea of what are possible key transcription factors involved in establishing or maintaining each of these cell fates. The next step would be, okay, what are then the target genes that are controlled by these transcription factors in these cell types? For that, we developed scenic a few years ago already. In scenic, in the first step, we derive modules of genes for each TF. So for each transcription factor is, let's say, on top, and a number of genes that are co-expressed with that TF. This can also be nonlinear. So this is using random forest regression to find these modules. Yields a number of modules of tightly co-expressed genes. And in the second step, scenic then analyzes the sequences around these genes per module. And for each module where the motif of the leading transcription factor is enriched in the sequences around the TSS of that co-expressed gene set, then we retain that module and we prune it further and call this a regulon. This then yields a set of regulons of TF and target genes that build a GRN. An example, so all these results are also for the fly cell atlas available in scope. So one could search for a TF and then observe or look at the expression of the TF, like glass is expressed in photoreceptors and in cone cells, but the regulon is active only in photoreceptors. And then you have the motif and the set of target genes that are predicted by scenic. So these are now key target genes predicted based on expression and on the motif of the transcription factor glass in photoreceptor cells. And for example, Ocellulus and Nina-C are expressed indeed in photoreceptors, and also in our spatial atlas, we can see very nice expression for Nina-C actually exclusively in photoreceptors in the retina, and for Ocellulus in the retina, and also in some more other cell types in the brain. Talking about the brain is a bit of a special case. So besides the fly cell atlas, our lab has also worked a lot on the fly brain. This has been led a lot by Jasper Janssens, who's now in Switzerland with Barbara Treutlein for a postdoc. And so the fly brain contains a few thousand different neuronal cell types, each with their own connectivity pattern and their own function, such as the Kenyan cells in the mushroom body, T45, here an example of optic lobe neurons or olfactory projection neurons. So besides the fly cell atlas, in my lab, we generated several versions of the brain atlas. Here's 250,000 cells. And for many of the cell types that have enough cells, let's say, like here, the Kenyan cells, we find clusters back in the single-cell RNA-seq atlas, or the olfactory projection neurons, or the T45 neurons. And also on that fly brain that's also published, we applied scenic to infer gene regulatory networks, and this is an example of a GRN predicted for olfactory projection neurons with key transcription factors, homothorax C15 and OTP, to regulate these predicted target genes. That gives us a GRN, a prediction, but often we were not really satisfied, and we wanted to go deeper into the not only the performance accuracy of the predictions of both TS and target genes, but also to understand more about the regulatory regions where these transcription factors are binding, so enhancers and promoters, and to understand better how these enhancers are encoded in the genome, what are their important features, and how do they really work, cooperatively or hierarchically, and so on, and which enhancers also work together, and how they control the promoter to regulate gene expression. And for that, single-cell chromatin accessibility comes to the rescue, single-cell ATAC-seq, and this is a method that we have been using a lot the last years to derive these candidate enhancers, the binding sites, and then build the eGRN now, the GRN with the enhancers inside from that. We developed several computational methods for that, also technologies. I will go over a few of these things, and we're applying in the lab these techniques of combined ATAC and RNA with computational methods to a number of biological systems. First, maybe a bit on the technology. So in ATAC-seq, we use TN5 to segment the chromatin. This gives us reads, genomic reads after mapping to the genome that are enriched for regions that are depleted for nucleosomes. And these nucleosome depleted regions also contain enhancers and promoters, and those are the regions that we want to decipher. This has been developed by Jason Penrostro in 2015 on the fluidigm, and since then, single-cell ATAC-seq got implemented on different platforms, such as SkyAttack on the combinatorial indexing and plates, or by Bio-Rad on the DD-seq or by 10x. And also in my lab, we developed our own custom method for single-cell ATAC-seq called HydroP-ATAC, for which we make our own microfluidic chips and our own hydrogel beads that are barcoded. And then we use this omics system with syringe pumps to generate droplets in which we encapsulate then a single nucleus that was previously segmented together with a bead. And then these barcodes on the bead are hybridized and linked with PCR to the ATAC fragments, so that after sequencing, we can demultiplex all the reads into individual droplets or individual cells. All these methods are in protocols.io. It's freely available. You can make your own chips, hydrogel beads, and it's of course much cheaper than 10x. Florian in the lab then, together with 14 different centers worldwide, led by Holger Heijn at CNAC, performed a benchmark study of all the different single-cell ATAC-seq methods, including HydroP. And as you can see, if after equilibrating the sequencing depth and even the number of cells, so this is all done on PBMC cells, human PBMC. If we take the same number of cells for cell types that have enough cells, all of the methods find the correct peak, also HydroP. But you can see that HydroP peaks are a little bit smaller than 10x if we use the same number of cells and the same sequencing depth. So HydroP finds the correct ATAC peaks, but it's a little bit less sensitive. This can be compensated on the one hand simply by sequencing more cells. This will also give you more fragments and higher peaks. For cell types that have too few cells, as we know, with the sparsity of single-cell ATAC-seq, actually none of the methods can really find the ATAC peaks. This is published last year, so you can go to the paper. There's a lot of benchmarking statistics to compare all these methods. HydroP basically works very well on the TSS and FRIP signal, but is much less sensitive compared to the commercial methods. In the meantime, we have made a version 2 of HydroP that increases the sensitivity. So this on the y-axis is the number of unique fragments we obtain per cell. And this is an important sensitivity measure. And actually, if we compare these red and green HydroP v2 with orange 10x v1, we are now in the second version of HydroP on par with the first version of the 10x, which gives us a very nice quality of single-cell ATAC data. In this benchmark study, Florian and Gert also made a pipeline called PUMATAC. It's available on GitHub that can take any single-cell ATAC technology as input and does all the pre-processing to identify the barcodes that are real cells, to get all the fragments, and to yield this matrix that basically contains all the single cells and all the regions in the genome that we include, with the binary, in this case, whether it's accessible or not in that cell. And then now we're going to continue on the computational side with that single-cell ATAC-seq matrix to build GRNs. And for that, we developed, this team here developed Scenic Plus, a recently published method that starts with the single-cell ATAC-seq data and performs topic modeling. So topic modeling comes from the text mining field. This is an iterative approach to learn that are basically, two probability distributions, one over the cells and one over the regions. So after this iterative optimization of these two probability distributions, this yields a matrix of all the cells over all the topics, so how much each cell likes a topic. And this is a limited number of topics, like 100 or 150 topics, and therefore it's a dimensionality reduction compared to the 100 or 200,000 of regions that you have. And secondly, and interestingly, we also have obtained from that a region topic probability matrix, where all the genomic regions, all the ATAC peaks basically have their probability towards each topic. And that matrix you can use to also cluster the regions, which then gives you a set of co-accessible regions over the whole atlas. And from these co-accessible regions, we are going to go further and investigate whether they're not only co-accessible, but whether the underlying reason for co-accessibility is actually co-regulation. And for example, that these set of co-accessible regions would have a shared combination of transcription factor binding sites, which would underlie their co-accessibility. The second step of Scenic Plus then is to take all these sets of co-accessible regions per topic into a motif discovery approach. For that, we use a ranking and recovery approach statistically that measures the area under the curve per motif and finds the motif that gives us the highest area under the curve. What motifs are we using? This is something that we have been, I started this in my PhD to collect all the position weight matrices in the world from all the different libraries. And now we're continuing curating that. We have more than 30,000 position weight matrices in the Scenic Plus database that are then clustered to find clusters of similar motifs. These are subclustered, and that yields then per transcription factor a set of position weight matrices. This is usually more than one because for most TFs, multiple techniques like SELEX or protein binding microarrays and so on have been used, or bacterial one hybrid, been used to determine these motifs, and they may vary a little bit. We still like to keep all these motifs per TF rather than defining an archetype motif. And we do that by using a hidden Markov model that basically uses every position weight matrix per transcription factor as a hidden state. And then we scan sequences for each TF using such a hidden Markov model. And then to test if this works, we used ENCODE data, so 309 transcription factor chipsec data sets. For each of them, we know the transcription factor that was chipped. And so the chip peaks could be analyzed for the motif, and we expect the chipped factor to be found as the first motif, of course. And that works the best here in red if we could contain all the position weight matrices and use multiple per TF in a hidden Markov model. And we find many more transcription factors at the first position of this motif discovery. So that's the technique that we use. After that step, we now have the attack peaks. We have candidate transcription factors, sometimes multiple per motif. And then in the next step, we use the single-cell RNA-seq data in an approach similar to Scenic, where we use GRM boost, so random forest regression, to link transcription factors to predicted target genes by expression, and also to link attack peaks to candidate target genes by a random forest regression or by correlation to say, okay, if the accessibility changes concordantly with the gene expression, then that deserves a link, an association with a certain weight. This then gives us TF to region, region to gene triplets, and these are all combined into a regulon, and all these regulons are combined into a GRN. In the paper of Scenic Plus, I will not go into detail. There's a lot of comparisons with other methods and benchmarks to verify or to test the sensitivity and the specificity of this approach. Let's go back for an example of Scenic Plus to the fly brain. We created a single-cell ATAC-seq atlas with 240,000 cells of the fly brain in the adult and during development. And so this is the adult data only. And the idea is that now per cell type or per cluster here, which are now the rows, we find a certain number of attack peaks. And some of them here in dark color are specific for that cluster. So this first row is the astrocytes in the fly brain. And there are a number of peaks like near wound 2 that are only accessible in astrocytes and not anywhere else in the whole atlas or in the whole brain here. And these are thus specific attack peaks for that cell type. All these co-accessible regions go to the motif discovery, as I explained. That prunes the set of regions to those that have a shared motif. And then we use the RNA-seq data to ask, okay, this candidate transcription factor through this motif, in this case, ILS here, PAC6, is found enriched. We look at the expression. And now every dot here is a cell type, and we find 3 cell types here, and these are all Kenyan cells that have high expression of ILS in the x-axis and that have high enrichment of the ILS motif in the attack peaks of these cell types. And that is, of course, when these two come together, we have more confidence in the TF because the expression correlates with the enrichment of its motif in the same cell type. And indeed, ILS is an important factor for the Kenyan cells. It has a mutant phenotype. It is well known for the mushroom body. This we do for all the cell types and all the TFs, and then you get here a list of TFs in the rows per cell type. And so you see that most cell types have some unique transcription factors that are now not only expressed in that cell type, but also have their motif enriched in the accessible peaks of that cell type. Some factors are pan-neuronal, some are pan-glial, and this gives us an overview of the TFs with the candidate regions and candidate target genes, as I explained by scenic plus. And so that gives us these EGRNs per cell type. We have them for about 40 cell types in the fly brain, where now in the middle are the transcription factors. And then you have here these nodes, these blue nodes, are the peaks, so the enhancer candidates, where the transcription factors are predicted to bind. And then these are linked to the target genes here in the red nodes, with the more red they are, the more expressed they are specifically in that cell type. So you see that most genes are actually linked to multiple enhancers. As you can see for this example, there are multiple attack peaks that are linked to this transcription start site, and that could regulate this gene. So here MEF2 and ILS are two key factors in Kenyan cells that bind to this enhancer and regulate this target gene. We could then also, because we're working in the fly, we have now the possibility to test these enhancers in transgenic flies. So we take 10 of these enhancers that are in the EGRN, clone them with a minimal promoter and GFP, make a transgenic fly, and then look in the brain and find, in this case, actually this worked extremely well. In 10 out of the 10 that we tested, the activity, the GFP here of the reporter is specific to the Kenyan cells in the mushroom body. So here you see the Kenyan cells, the cell bodies, and the processes. Here we use GAL4, so you also see the processes nicely recapitulating for all these 10 enhancers. That was already working well. Still, we wanted to go deeper and further into the enhancer sequence and try to learn more about how does it really work. And for that, we're using deep learning models. So enhancer models, state of the art at the moment, is using convolutional neural networks, where you start with a convolutional layer, basically with filters that scan over the sequence. These filters are of the size, the width of a transcription factor recognition site. And so you basically scan over all these sequences with all these filters, and then additional convolutional layers are able to capture more complicated aspects of the syntax and then predict for each sequence as input, predict, in this case, the accessibility. That's how we use it mostly, but you can also predict activity of the enhancers depending on the data that you use for training. So this sequence here is accessible in 2 cell types, and the model is trained to predict that is accessible in these 2 cell types. Then you get a model to predict attack-seq. It's nice, but we already have measured attack, of course. But the power for these models really lies in the possibility, I will go back to this, in the possibility to then ask, why does the model predict this sequence to be accessible in that particular cell type? What are the input features? Which nucleotides have contributed most to this prediction? For that, we use methods like SHAP and Deep Explainer, or in silico mutagenesis, as shown here. I will come to these examples later on to highlight the important nucleotides. And this is really where the power is. This allows us to identify the transcription factor binding sites in an enhancer and to do a lot more cool stuff. So these type of approaches are used in the field, training on DNase hypersensitivity, training on bulk attack-seq. So DNAs like DeepMind, DeepC, from BPNet, from the Kundaje lab, trained on TF binding, like BPNet, trained on enhancer activity data, like DeepStar from Alexander Stark's lab, or trained on single-cell attack-seq. That's what we usually use. And these are some of our models that we published, Deep Flybrain, Deep Liver, Deep Cortex, or even trained on, with larger input spaces, on gene expression data to also predict gene expression. That is done by Informer and Basenji from DeepMind and Calico Alphabet companies. So an example here on the Flybrain, Ibrahim trained this model, convolutional neural network, in this case also with an LSTM layer. In the most recent models, we don't use these recurrent layers anymore, but this is also interesting. And we trained this model on the single-cell attack-seq data of the Flybrain to predict here, it's a classifier, to predict for each sequence to which topic it belongs. So the topics from the single-cell attack, remember, here are some examples. You can have a panglial topic, a pan-neuronal topic, or topics that are specific for each cell type. And so each region has a topic probability vector, and so we can predict that based on the sequence, and the model is trained for that. After it is trained and it is shown to work on performance, on test data, then we can start using the model to explain some of our predicted enhancers. So this is an enhancer we predict for T4 neurons in the brain, and the model suggests these nucleotides to be important, and they agree with transcription factors that are important for T4 neurons like FORCAT, TFOP, and ACJ6. We could then also clone this region that is predicted by the model, like I showed before, with a minimal enhancer and a nuclear GFP in this case, make a transgenic fly and test, and indeed, this sequence is indeed specifically active in T4 neurons. This is a Kenyan cell enhancer with predicted ILS and MEF2 binding sites, and it is also active in the Kenyan cell specifically. We use this model to predict many more Kenyan cell enhancers, and we have, again, a very high success rate of genomic enhancers that have such a code, and the model predicts to be Kenyan cell specific. If we test it, it's indeed specific, as you see here, for all of them in Kenyan cells. By collecting all these motifs, also called seclets, across hundreds of enhancers, we could build what's called code tables, where you see the different patterns that are important per cell type. So alpha beta Kenyan cells, gamma Kenyan cell, alpha prime, beta prime Kenyan cell are 3 different types of Kenyan cells. And for all of them, one-cut ILS-MF2 is important. Their motifs are important, as you can see. And also this ball here is the expression of the TF. So in alpha prime, beta prime, MEF2 motif is a bit less important, but also the expression of the factor is less. So that goes together, and you see it even more clearly in the optic lobe T neuron cell types, where FORCAT is expressed in all these T neurons, but not in T1 and T2. And that's exactly where the pattern is important. ACJ6 is important everywhere except in T1, and that's also not where it is expressed, so the pattern is not used by the model. So this gives us a lot of confidence. We also find, and this will come back also later in the talk, repressor motifs. So these are motifs that the model uses to predict that the region is not accessible. So when this MAMO motif occurs in a sequence, it's predicted to be not accessible, and that agrees also with the expression of the TF. So MAMO is expressed in these 2 cell types, in the Kenyan cells, and there it is predicted to act as a repressor of chromatin accessibility. So we see that the regions that have this motif EIP93, for example, in alpha beta are not accessible in alpha beta. Next step, what we could do is in silico mutagenesis for each enhancer, mutate every base pair, every nucleotide to every other nucleotide, and plot here in the y-axis the delta prediction score. So these nucleotides here are predicted by the model to decrease the enhancer accessibility and predicted also to decrease the activity in that sense. And we tested them by including those mutations in the sequence, making new transgenic flies, and indeed they abolish the enhancer activity. And these mutations here are predicted to increase the enhancer accessibility because they mutate this MAMO motif. And indeed, in vivo, by testing this enhancer in the transgenic line, it increases the enhancer activity. So we thought this model seems to capture very well the Kenyan cell and other glial types and also T neuron enhancer code. And you can basically then annotate or enrich your gene regulatory network by these deep explainer scores. So this enhancer here is an attack peak in 2 of the 3 Kenyan cell subtypes, and this is exactly the 2 where the target gene SMPF here is expressed. So these 2 Kenyan cell subtypes, but not the 3rd one, alpha beta prime. It's not expressed. It's also not accessible, and this region is working as an enhancer, as we can show by a transgenic fly, and then the deep learning model can give us the information on which motifs are binding there. So after this model was evaluated and validated, and we were very happy with it, Ibrahim wanted to use it to take a next step and to try to make synthetic enhancers using this and other models. And for that, we're using the models, basically these deep learning models, as an oracle in an optimization algorithm, a state space search, if you like. We start from a random sequence and mutate every position, score all these mutated sequences with the deep learning model, and choose the mutation that gives us the largest increase in prediction score for the targeted cell type. Then we take that mutation and then we mutate everything again in a second step, a third, and so on. So this is an evolutionary process that starts from a random sequence and tries to find the optimal path in a state space towards a local optimum, and that has a high score according to the model, and that should be an enhancer. A second strategy we used is to implant the motifs, because we already have them from the code tables. We can implant the motif at every position in a random sequence, use the model to score all of them, and select the position that has the highest score, and then insert a second motif and a third motif. And the third strategy we used are generative adversarial networks, or GANs, that are trained to spit out sequences, to generate sequences that can no longer be discriminated by the discriminator from real enhancers. In each of these strategies, we can design an enhancer, synthesize it, or send it out to a company to synthesize it as a sequence, and then clone it with a minimal promoter and GFP and see if these sequences that are designed by the AI model are actually active in the fly to drive a GFP. So we start from thousands of random sequences and could observe that after 10 mutations already, the majority of these random sequences obtain a prediction score by the model that is as high as the genomic enhancers. And so that you only need relatively few mutations from a random sequence. We also observe that these mutations, these 10 mutations, are usually confined within a 200 base pair window, while we give 500 base pair room, arguing or suggesting that enhancers are usually of that size of 200, 250 base pairs, which actually is an NDR, or nucleosome depleted region size. So we chose some of these synthetic sequences. You can see an example here. A random sequence has a very bad score. It is full of these repressor motifs. And then you can see every position here that is mutated and their sequence. So the number 1 mutation that's chosen here destroys this repressor motif. The number 2 mutation destroys this repressor motif and actually also generates an ILS motif. Then other mutations make this ILS motif better, create a one-cut motif, another ILS motif, and a MEF2 motif. After 10 mutations, this random sequence is converted into a full-blown enhancer according to the model. We can then test this, and indeed, this enhancer works beautifully specific in Kenyan cells, showing that it is really indeed a Kenyan cell enhancer. A few of them were negative after 10 mutations, so we didn't really see a GFP activity. Then Ibrahim took those sequences back, include 5 more mutations, and now they become positive. So sometimes you need a few more mutations, and you can still make them to positive enhancers. And some of these are really exquisitely specific and very, very strong enhancers that do not exist in nature, but that can drive GFP specifically in Kenyan cells in the fly brain. Interestingly, we also found regions in the genome of the fly that are, we call them near enhancers. They already have a high score, but they are not really accessible. And we found that these actually require only few mutations. And after 6 mutations, they can become already an enhancer. So this argues that in evolution, it is not so difficult to make, in this case, Kenyan cell enhancers. And some regions in the genome are already almost a Kenyan cell enhancer. And with a few mutations, they would become active. We did the same. All of this, again, also on another cell type, pyreneural glia. We have code tables. We designed synthetic enhancers. Now, starting from the same random sequences, so this is interesting. Ibrahim used the same random sequences as for Kenyan cells. 10 mutations make a Kenyan cell enhancer. 10 mutations, in this case, 15 mutations for glial cells make a glial cell enhancer. And we also make transgenic flies for these, and they work beautifully specific in pyreneural glia. And this shows, because we start from the same random sequence, it shows that the information that the enhancer uses to get to this activity is only in the positions where we did the mutations and not in the flanking sequences. And every mutation that the model made is interpretable as either generating an activator motif, making an activator motif stronger, or destroying a repressor motif. And that argues that these enhancers for this system and also for other systems that we have tested can be defined by the right combination of transcription factor motifs, their strengths, and their arrangement. Because if you start moving the motifs, then also you can break the enhancer if it is not the right arrangement anymore, and the present and absent of repressor motifs. And these are the key criteria to make an enhancer according to this work. Second strategy, quickly, we also made them by motif insertions, show that if you insert ILS or MEF2, doesn't really work. But if you insert an ILS and a MEF2, it really has a high score already. If you do that across thousands of sequences, you can actually calculate what is the optimal distance between two motifs. So 5 base pairs is actually enough between MEF2 and ILS, and 3 base pairs is enough between one cut and ILS. And that led us to generate enhancers that we call minimal enhancers that are only, in this case, 41 base pairs with 3 motifs and with these minimal distances in between, and this works as well. We included some 4 base pairs on each side also as a flank. 49 base pairs works as well and as specific as the 500 base pair sequence, showing that in this case, yeah, enhancers can actually be created only using motifs because there is nothing else in this sequence. By the way, if we take this minimal enhancer and ask, can the model evolve this now to break it? What do you have to do to make this enhancer inactive without allowing it to touch the activator motif? So can we destroy the activity, but we cannot mutate the activator motifs? And then the model suggests to do 1, 2, 3, 4, 5 mutations. Each of them, this one is in between these 2 activator motifs, each of them generating a repressor motif. And by testing this enhancer, it indeed doesn't work anymore. So this shows that the repressor motifs can indeed be dominant and overrule, even though this is a beautiful combination of activators by themselves, it works beautifully, the repressors in the neighborhood can cause this enhancer to not work anymore, showing again the importance of the balance between the repressor motif and activator. And this is also why we think that earlier efforts to make synthetic enhancers didn't work by just implanting activator motifs, because you don't know in your background random sequence whether there are no repressor motifs present. And this could maybe be the reason why some of these efforts have not worked. We could do a number of interesting other stuff like generating enhancers that, or here, this is an answer that works in the genome in 2 cell types, and we could use a model to prune it to work only in 1. So we're able to mutate exactly those positions that abolishes 1 cell type, but not the other, or reciprocally, an answer that works in 1 cell type. We could add another cell type into the same sequence, and this enhancer is not active in Kenyan cells, but it is in T neurons. After the mutations, it remains active in T neurons and now gains activity in the Kenyan cells. And this is all in a sequence of 200-300 base pairs. So you can actually put 2 enhancer codes into 1 nucleosome depleted region. That's also new. For human enhancers, this works in a similar way. We used a model called DeepMel that we trained on ataxic data of human melanoma samples that fall into 2 states, mel and mes, melanocytic. So these look a lot like melanocytes, or mesenchymal, and then use this model to predict enhancers and to design enhancers. And this also gives us an information on the binding sites of transcription factors in a melanocytic enhancer. And we could actually show that the model can predict which nucleotides are important. And by testing them all in a massively parallel reporter assay, this is from the published paper, in vitro, this shows a very high correlation between the in vitro measurement. Making this mutation causes the enhancer to be less active, and the model indeed also predicts that. And so most of these positions agree. You can make the enhancer less active by introducing such point mutations. The model agrees with that. The in vitro data also. Each of these actually generates, again, one of these repressor motifs. In this case, these are ZEP motifs. And by using other architectures, other deep learning frameworks, we could actually confirm that these indeed are repressor sites. And this actually these 2 models actually agree very well on the predictions. By mutating this repressor site, we could show that now the enhancer activity by luciferase goes up. And by adding more repressor sites, the enhancer activity goes down. Similarly, as in the fly, also in human, the balance of the repressor motif is important. And indeed, by ChIP-seq, we could show that this ZEP2 is the factor that actually binds to this enhancer together with the activator. So there's a kind of a balance happening here. We don't know if it's on the same allele, of course, on the same time point, but this balance really occurs. This model we also used to generate synthetic enhancers. So a random sequence is converted after 15 mutations to a full-blown melanoma enhancer for the melanocyte state. And you can see the different steps. Again, activator motifs are created, repressors are destroyed, additional activator motifs are created, and each time we increase the enhancer activity here from a certain point onwards, it starts to catch, and then you can make it also stronger. I will skip this because I'm running out of time, because I wanted to discuss a little bit more of some unpublished work where we are now using these principles and these models to test whether we can use them to compare different species. And this may also be relevant to the ERGA, where genomes of different species are compared. can we use them to compare cell types in evolution? So basically, we are moving this whole framework from the fly also to vertebrates and mammals. This is a chicken brain, mouse brain, macaque brain, in the idea of studying GRN evolution. So from the Eric Davidson era, it was already suggested that key differences between species are in large attributed to changes in gene regulatory networks, whereby even the same transcription factors could be conserved, and we often see that in cell atlases that between species that transcription factors are often the most conserved genes, but that downstream certain genes are no longer in the network. Other genes are added to the network in species B compared to species A, and that is due to the turnover of the enhancers and of the binding sites. So between two species that are quite divergent, like birds and mammals, nearly like almost none of the enhancers of human can actually be mapped or aligned or lift over to the bird genome. Most of the enhancers have turned over. Most of the binding sites have turned over. New enhancers have been generated. Nevertheless, if the transcription factor combination is conserved, that would mean that the code would still be conserved. And even though these instances of enhancers are different, they may adhere to the same model. Meaning, if we train a model on the enhancer code of species A, like deep flybrain and deep mel, and use them to score sequences in species B, we can maybe track orthologous, let's say, code enhancers. We tested this approach, Nikolai and Niklas. We are writing a manuscript about this. It's not ready yet, it's in preparation, on several brain data sets. So we used 2 mouse data sets of single-cell multi-omics, so attack RNA of the mouse cortex, and 2 brain data sets of the human cortex. The cell types between human and mouse cortex, as shown by the Allen Institute, are one-on-one, let's say, orthologous. So for each of these layers in the mouse and in the human cortex, we have corresponding cell types, as was shown already in the papers by the Allen Institute. So all of these cell types listed here, non-neuronal, glutamatergic, and GABAergic neurons, have one-on-one matches between human and mouse at this subclass level. So that means we could compare the enhancers between these for each of these cell types. This is an example for microglia. So this region here in CSF1R in an intron is a region that is only accessible in microglia, not anywhere else. And the gene CSF1R is actually a marker gene of microglia. So we can take this region, it's actually a validated enhancer of microglia in mouse. So this is the mouse sequence that we see. And we interpret it, we give it to the, by deep explainer, to the different models that we train. So 2 mouse models and 2 human models. And what you can see is that these 4 models, by and large, actually agree on the importance of the motifs in this sequence. So not only does it confirm the 2 different mouse models, but also the fact that the human and mouse are interpreting this sequence in the same way, arguing for a conserved trans environment. The transcription factor combination in human and mouse is likely conserved because they interpret the mouse enhancer in the same way. This would be the idea. We could further discuss that too if you have questions about this. To make it more interesting, we use a species with some divergent cell types. We use here a juvenile chick brain, where we perform single-cell multi-omics on the entire telencephalon and on a slice through the telencephalon. This is the multi-omic data. We find all the non-neuronal cell types back very easily: microglia, astrocytes, oligodendrocytes, OPC, and we have also glutamatergic neurons and GABAergic neurons. Because we know less about the bird brain, we performed spatial transcriptomics also on this same, on a consecutive slice, in this case by stereosec, and use cell-to-location to map these clusters onto the spatial data, and we find a very nice localization of these different glutamatergic cell types, as you can see here in more detail. So GLU1 and GLU2 clusters here map here to the hyperanidopallium, GLU3 specifically to the endopallium, and then GLU4, 5, and 7 map to the mesopallium, and 6 and 8 map to the hippocampus. And this way we already get an idea of what type of glutamatergic neurons they are. Then the first comparison we can do is on the transcriptome, comparing all the chicken transcriptomes with the mouse transcriptomes using SumUp. And for the non-neuronal cell types and for the interneurons, we find one-on-one matches. But for the glutamatergic neurons, as expected, because birds don't have this layered cortex, we have more trouble linking these different clusters. We have 8 glutamatergic clusters with the different glutamatergic cell types of the mouse. Nevertheless, there are already some hints here. Some cell type like GLU7 seems to match better with layer 6 CT, and we wonder if this could be real. And for that, we're going to go now to our enhancer models and see if we can have support at the underlying GRN, at the regulatory code for this transcriptome resemblance. This was actually one of the best matches, GLU7 with layer 6 CT. So on the single cell attack data of the chicken brain, we trained again this CNN, the convolutional neural network, like we have for human and mouse. All right, and then we use it. In this case, first evaluate the mouse microglia enhancer. You see here the chicken prediction of the important nucleotides, again, matching extremely well with the human and the mouse, arguing that also in chicken, microglia transcription factor combination is conserved. Now we can go to more difficult cell types and do it for all cell types. Take for each cluster in the chicken brain all the attack peaks and present the sequences of those peaks to the mouse model. So we're going to give them as input to the mouse model, and it will predict the mouse cell type for a chicken region. As you can see, for the microglia, for astrocytes, for oligodendrocytes, we have one-on-one matches. So attack peaks for microglia score highest for mouse microglia. Attack peaks for astrocytes are scored by the mouse model for mouse astrocytes. And this actually then allows us to annotate the single cell attack data using the mouse model. For glutamatergic neurons, again, it is more diversified as expected, but we also see some resemblances. Again, GLU7 seems to match with layer 6 CT, one of the highest matches, similar to the transcriptome. 6 and 8 match better to the hippocampus, which we also had from the transcriptome. So this does seem to hold. A second strategy we use is to compare the deep explainer profiles between two models on the same sequence. So we use a chicken sequence, explained by the chicken model, and explained by the mouse model. For the mouse model, we can do this for every mouse cell type and ask with which cell type is the correlation highest. And then we find that for chicken astrocyte regions, the mouse astrocyte class gives us the highest correlation. For microglia, it's microglia. For GLU7, it's again layer 6 CT, and for GLU1, it's again layer 2-3. So you see there's a strong correlation for some of these regions between mouse and chicken. For example, astrocyte, there's this mouse region, only accessible astrocyte. Chicken, same gene, well, orthologous gene, orthologous region. Actually, there's a lift over between these two, only accessible in chicken. And if you explain them by the different models, then you see that they are highly conserved in regulatory code. And this also is a conserved sequence. And you can see in red the mutated sequences between the genomes of chicken and mouse. And actually, they avoid all the important nucleotides. So you see here two mutations right in the middle between this dimer motif here again. And the important nucleotides are not mutated between birds and mammals. Again, kind of validating that the predictions of our model that these nucleotides are important is likely going to be true because they are under negative selection. If we look at the code table between mouse and chicken for astrocyte, OPC, oligodendrocyte, microglia, we see a very strong correlation. So the same patterns are used to make these enhancers between these vertebrates. A last strategy I will skip. There's not much time. This is the comparison of the code tables, but I will come to this. We use 3 different strategies to compare enhancer codes and one to compare transcriptomes, which is sum up, and then put them all together in this pie chart, if you like, pie chart heat map. The larger the circle, the larger the correspondence, and you see that now astrocytes link with astrocytes between chicken and mouse for transcriptome and for the enhancer code using 3 different strategies. OPC also, medium spiny neurons, D1 and D2 are difficult to distinguish. The interneurons, and then here in the glutamatergic neurons, you see that all glutamatergic enhancer codes are corresponding to the human and mouse glutamatergic, so glue matched to glue. But within them, there is more diversification, except for GLU7 and GLU1, for which we think the scores are high, maybe even here GLU2, high enough to potentially argue for either an ancestral common ancestor that already uses this regulatory code or a co-option of the same combination of transcription factors. That is, of course, without a phylogenetic tree, always hard to argue. So we find correspondences between the layers of the mouse cortex and the nuclei of the bird brain. With that, I want to conclude just one slide. If you're interested, if you use Minecraft and if you have children and you may be interested in a module we developed in the lab called Mendelcraft, this is a mod for Minecraft where you have a lab and a sequencer and a bioanalyzer, and you can use the DNA of chickens and analyze them in the lab to cross, but also to change and edit the DNA in an egg and to clone chickens and so on, and to investigate and teach children about the rules of Mendel. And if you find the right alleles, you can even make a dinosaur out of a chicken egg. And we've been doing lots of these workshops. We have made YouTube videos and a website, so it's easy to port. There's a book inside Minecraft, so if you want to distribute that in with your children, then be my guest. I want to thank you all again for coming, for listening, for the invitation. Thanks everybody of the lab, our funding, our collaborators, and I'm very happy to answer any questions. And thank you so much.
Well, thank you, Stein. That was incredible. There's a couple of questions that have been copied to the chat that I think you can also see. The first question comes from Danny Huilebroek.
Hi, Danny.
Are the near enhancers similar to those that Pinaccio and colleagues name as shadow or hidden enhancers?
No, they're slightly different to me. So shadow enhancers are occurring a lot also in the human genome and mouse genome, in the sense that one gene has multiple enhancers that adhere, let's say, to the same enhancer code. So they would all score high, 4 or 5 enhancers score high for a particular class. If you would investigate them, they would all have similar combination of transcription factors, and they would actually all also work in a reporter assay. How they work together to regulate the target gene is still a little bit under study. This could be in the shadow enhancer, it's argued for redundancy. It could also mean other additive or multiplicative functions, while the near enhancers are sequences that are not an enhancer. So they are in our genome. There are sequences that already have some score for the prediction model, but if you test them, they don't work. And they already have some binding sites, but there's not a full-blown enhancer. Maybe there's too much repressor sites. They become a full-blown enhancer with fewer mutations that you would need from a random sequence. That was the near enhancer. I hope that's clear. Danny likes the Z multitude scores. Yeah, Danny is world famous for his work on Z transcription factors. Shown as half sites. Have you looked at the B part? No, we have not looked at an 85 base pair. We only looked at the sites that the model recurrently finds. So this is data-driven. This is not something we impose. So we let the model across all the enhancers come back with the clustered seclets, which are called patterns. And we find the ZEP pattern there as a repressive one, but we don't find larger one. 85 is also very large. So our convolutional filters are only 22 or 24 nucleotides long. So we don't find these long motifs. Then Christian asks, could the synthetic enhancer have some kind of epistatic effect? If yes, how can you account for them in more complex brains? You mean that it would have activity in multiple cell types, or what do you mean? Or that it would be false negative, so it would have some kind of a... Could you explain this more? Multiple cell types, yes, this multiple cell type, yeah. Yes, but then we expect this to be present also in the training data. So if two cell, like I showed for the Drosophila and optic lobe, we had enhancers that work in 2 cell types, but they were also accessible in these 2 cell types. So the model has learned that if you're accessible in these 2 cell types, you have to predict that as to accommodate the sequence, also to learn the sequence, how the sequence can accommodate these 2 cell types. And we found out that in those cases that we studied, it happened to be that these two codes were independent of each other even, and we could manipulate them independently. We also found examples where the codes are overlapping, so one binding site would be shared between two cell types, not necessarily by the same TF. So let's say a homeobox transcription factor motif, the homeobox could be bound by a homeodomain factor in one cell type and by another homeodomain factor in another cell type, and there would still be overlapping codes. Yeah. Then Lara, sure, it opens many possibilities for new treatments of brain tumors. Yeah, enhancers as targets. Yes, excellent idea. We are thinking about this too. Also talking to some companies for designing enhancers for specific cell types for gene therapy. Because if we already know a vulnerable cell type and we already have ideas of transgenes that we could express to basically bring them back or to kill them or to bring them back to normal, then we will still need an enhancer that is specific for that cell type. Because for gene therapy, delivery with AAV, for example, would infect many cell types, would infect many cells in your body. And one key aspect for safety would be to specifically express the transgene only in a cell type of a target. And so we need those enhancers. And I think our approaches are well suited to design them. And we are also talking to some companies to do that with them. So I think it will be a nice feature. Now it's also published, of course. So yeah, I hope other people in the world are also going to use this for potentially enhancer therapies. Yeah.
Cool. I also have a question, if I may. I'm really surprised about the low number of mutations you need to go from a random sequence to an enhancer. But then in the chicken and mouse comparison, it does seem like you find a good amount of homology between the enhancers. And I just wonder what your expectations would be on evolutionary scales to find this homology or this regulatory logic.
I see. Yeah, okay, yes. Maybe this is a bit misleading indeed. So I showed you one example of an enhancer in astrocytes of chicken and mouse for which we actually find the enhancer back by liftover. So a whole genome alignment actually, yeah, finds this back. So this enhancer is actually conserved. The nucleotides that did change fall outside of the binding site. So this is all fine. Now, this is an exception. So in fact, the majority of the human enhancers do not have a liftover sequence to bird genomes, to the chicken genome. So showing that most of these enhancers have changed so much that there's no sequence alignment anymore. All right, so the turnover is really high compared to protein coding genes. Yeah, you can find your orthologous genes back in the chicken genome, right? You do a reciprocal blast, you find your genes back. Those are under strong negative selection. The enhancers are under much less negative selection. Some are, we don't know why, what is special about the conserved ones, but the majority are not alignable anymore. Nevertheless, they can still have the same code, the same combination of transcription factor motifs somewhere else around this gene. So this again also, this is an agreement because it shows that enhancers are a playground for evolution. There's a lot of changes between species. A lot of enhancers are different. And still, you have those enhancers. So you can actually destroy and generate them quite easily. You see, so that goes together with the fact that you don't need that many mutations to make it to get a de novo enhancer. It is not that complicated, right? If you need 5 or 6 or 7 mutations, yeah, this can happen in evolution if we compare birds and mammals, right? This is a bit the reasoning. Of course, yeah, it's also speculation.
Yeah, no, it's interesting. I wonder about this idea of comparing phylogenetic footprinting, traditional techniques of searching for homology with the regulatory logic and see how the two fit. Yeah.
The former one with the footprinting is limited to more closely related species. That I can already say. If you want to do that between human and fish, there are hardly any enhancers that you can map to fish. So you don't have any possibility to find negative selection within a mutated, like let's say a conserved sequence, and then go figure out what the binding sites are. This works well in relatively closely related species, from human to mouse, for example. Yeah.
Yep. Okay, I see no more questions in the chat, and we're also at time. So Stein, thanks again so much for giving this presentation.
It was a pleasure. Thank you, Steven, for the invitation, and everybody from the ERGA Consortium and the listeners online. Bye-bye.